Hi, my name is Seb James. I'm a postdoc in Stuart Wilson's group at the University of Sheffield. The development of the axonal projection from the retina to the tectum is an important model for the study of how the brain organises its connectivity during development. In fish, amphibians, and mammals, retinal ganglion cells grow their axons into the brain and make topographically ordered connections on the tectum. Computational models of this system first appeared in the 1970s, but most of these have been phenomenological models, some using mechanisms only possible with a global supervisor. I wanted to find out whether a self-organizing model could be found operating with plausible signaling mechanisms that would describe both the normal development of the projection and its behavior under experimental manipulations. I started with Simpson and Goodhill's model, which provided an explanation of the wild-type retinotectal development and also accounted for the results of surgical and genetic manipulations. The movement M of each axonal growth cone is driven by the sum of three effects: chemoaffinity, axon-axon competition, and a receptor-receptor interaction. The model invokes a global supervisor which takes on chemoaffinity's role of guiding each growth cone to its target. Similarly, the competition effect provides no physical mechanism that would induce the repulsion of axons away from each other. Only the receptor-receptor interaction invokes a signaling mechanism. I sought to modify the Simpson and Goodhill model by replacing the global supervisor chemoaffinity with a gradient-following model, and by investigating whether a single axon-axon interaction could provide the competition. To implement signaling mechanisms, we must define the expression of the molecules that achieve the signaling. In 1995, retinal axons were shown to be guided along the rostral caudal axis on the tectum by the ephrin A family of receptors and ligands. Other ligands and receptors have subsequently been shown to participate in retinotectal mapping, and in this work we imagine that 4 pairs of receptors and ligands are involved, 2 pairs per spatial axis. The maps in B to D show the expression of simulated receptors and ligands on the retina and tectum. Our retinal receptors R0 and R1 have the colours red and green and are expressed in orthogonal directions. 2 more receptors, expressed in opposing directions, are shown in cyan and magenta. These retinal receptor expression values are carried by the axons as they grow into the tectum, where they interact with the tectal ligand gradients of their ligand partners to give the chemoaffinity effect. Axons may also interact with each other via receptor-receptor signaling or receptor-ligand signaling. Definitions of the terms used in the equations can be found in the slide notes. Gradient following, in which axonal growth cones climb or descend gradients in the expression of a signaling molecule, needs a stopping mechanism. Something needs to prevent the growth cones from flowing off the edge of the tissue or gathering up along its edges. One solution is to add a counter gradient with a variable interaction of each growth cone branch with the 2 counter gradients. This can preserve the topography of the arrangement. Using expressions from the literature for receptor expression in a model with only gradient following, we find that the topographic arrangement is preserved in comparison to the expected experimental result, but the scaling of the map is not necessarily correct. Evolution may have adjusted the receptor and ligand expression gradients in order to produce a map which is both topographically arranged and scaled across the entire tectal area, as shown in this model. However, the gradient-only model is not able to reproduce experimental manipulations shown here. It is especially bad at reproducing certain ablation manipulations, such as retinal ablation, where it is observed that axons from an ablated retina will grow into a map which extends across the entire tectum. However, the feature of opposing gradients in this model mean that a single axon will find its way to its target location, reproducing the experimental result of Gosser et al. 2008, in which competitive effects were genetically disabled, and which is often cited as an argument against the importance of or need for competitive effects in retinotectal mapping. Competition as a mechanism for axon guidance was discussed by Prestige and Wilshaw in a computational model of the retinotectal projection as early as 1975. In our model and in Simpson and Goodhill's, competition is cast as a tendency for axonal growth cones to avoid getting too close to each other. Simpson and Goodhill modelled a mechanism by which growth cones know, by a global supervisor, when they are close together and are repelled along vectors that will drive them further apart. Here, we see that chemoaffinity, with the gradients that would result in the contracted map, combined with competition, leads to a topographically organized map that spans the tectum. This slide shows the results of the chemoaffinity plus competition model. Its three parameters, gradient strength, competition strength, and competition range, were obtained from a simulated annealing optimization of the layout error of the wild-type model. Competitive repulsion ensures that axons extend across the tectum in the retinal, tectal, and mismatch ablation manipulations. In graft simulations, ligand expression levels are carried with the graft, and the resulting discontinuous gradients lead to map modifications that are qualitatively similar to those observed in surgical experiments. The model reproduces some of the qualities of gene manipulation experiments by Brown et al. And Reber et al., although it does not reproduce the feature of map collapse described in Reber et al. The last idea I want to talk about in this presentation is what might be the origin of the growth cone repulsion effect. To produce the results shown on this slide, I replaced the globally supervised competitive repulsion with a receptor-receptor signaled repulsion of the type described in the Simpson and Goodhill paper. In contrast, I adopted this receptor-receptor interaction for all four receptor types in the model, whereas Simpson and Goodhill limited their treatment to the FA receptor expression and retained the generic competition. I carried out the same parameter search for the wild-type system. The results obtained in the simulated manipulations are very similar to those for the generic competition. To summarize, a purely self-organizing gradient-following model can reproduce the same set of qualitative results as the globally supervised model of Simpson and Goodhill. A single signaling mechanism for axon-axon interactions appears to be sufficient to reproduce the results of the earlier study. This model provides a platform to investigate the effects of noise on gradient-following models. The noisy sampling of tectal ligand expression due to noisy receptor binding has been raised as a problem with gradient-following models. I'd like to acknowledge the financial support of the McDonald Foundation, which funded this work.